And actually, the development of that drug was greatly facilitated by having mouse models that overexpressed the EGFR gene, so that you can then develop a mouse tumor that replicated that particular feature of the human spectrum of genetic mutations. I think also the topic that I just covered at the end about cancer stem cells has certainly elicited an awful lot of new discussion about how you might specifically target those tumor stem cells. So maybe that needs another 5 or 10 years to actually get to the clinic, but it's actually quite an interesting idea and potentially promising.
Yes, I have a question, and my question is that would you please explain that how this model, which is in a controlled environment, will be, give you opportunity to explore or explain the gene and environmental interaction?
Okay, that is a very good point. What we're trying to do by controlling the environment and the genetic composition of our models is very dissimilar to what happens in a human population. We don't really control what a human population is exposed to, and actually it's very hard to even account for what a human population is exposed to. If I ask you if you ate any chocolate last week, half of you would admit it and half of you would deny it. It's just one of the things about studying humans, there's a lot of variability at lots of different levels. So in experimental models, what we try to do is control all those factors. We feed the mice specific diets. We control how much water they drink. We can control how much light they're exposed to. And by doing so, we reduce the variability within our experiment, but we sacrifice the diversity that really represents humans. So that's, again, one of the trade-offs that comes with being an experimentalist. You have to, if I did an experiment similar to what essentially is a human population, I would need so many mice to actually come to any conclusion that nobody would ever fund me for it. And so for this, we make those decisions just as a trade-off.
Yes, how similar are the mouse models to the human condition? Could you review the similarities and differences?
Well, I think that they're similar in that they both develop postnatally. They both require ovarian hormones in order for that development to occur. That we see in both cases a genetic susceptibility that we just discussed that influences profoundly whether or not you get a population in which 1 out of 3 individuals develop cancer, or one where 1 out of 13 develop cancer. So that genetic composition is there. Furthermore, they're similar in that the lifetime events modify cancer risk. So exposure to external agents or undergoing pregnancy. In humans, we know that an early first pregnancy confers a lifetime protection, decreased risk of developing breast cancer. We still actually don't understand why that is. The only way we're going to be able to address it is by using mouse or rat models, and interestingly, both of them show the same characteristics. If you induce pregnancy early in a rat or a mouse and then expose it to a carcinogen, you get much reduced cancers in those individuals, even though you've controlled very carefully that they are exposed to exactly the same amount of carcinogen. So I think by using those similarities, we actually can advance our knowledge of why it is that these lifetime events in humans have such a profound effect.
Could you give us a couple of key points from your talk today, and then discuss a little bit about how we could translate this information into something that is useful to the general public?
Well, I think the major point that I tried to address today is why we use something as dissimilar from humans as a mouse to study something as fundamental as breast cancer. And what I hope to impress upon everyone is that there's actually a number of different levels of similarity that we can, again, choose to study in that particular context. In science, we often, almost invariably, are dealing with models. We can't and won't use humans to study questions like this. How do you cause cancer? We're not going to go out and expose a new population of women to, for example, a detrimental agent like ionizing radiation just so we can get a better assessment of who is susceptible. So instead, we have to create models that will allow us to dissect not only the frequency of an event, but actually the contributing causes to that event. Be able to dissect down the cells and be able to from the cells to the molecular events that occur to generate a cancer. So the idea here was really just to discuss why has NIEHS decided to fund essentially 4 different centers across the country who on the one hand are studying determinants of puberty in young girls, and on the other hand are studying mouse mammary glands. Actually, some of them are studying rats, so rodent mammary glands. It seems like an odd mixture of questions to have. And the idea here is that only by combining the human studies with the mouse studies will we actually be able to move forward. Now, how is that going to be implied? Well, that's an interesting problem. Sometimes scientists have a hard time actually addressing that because they're involved in the immediacy of, the next experiment and the next proposal and the next paper. But ultimately, the goal of this project, which is actually funded for a period of 7 years, is that after this interaction between these 2 different projects over the course of this next 7 years, we'll have a better handle on what questions we should be asking in the human case, right? And which questions we should be studying in the mouse models. So I'd like to end by acknowledging a variety of people who've contributed to the production. First of all, this work was supported by the National Institutes of Environmental Health Sciences and the National Cancer Institute, which provided the funding for the Bay Area Breast Cancer and Environment Research Center, which Bob Hyatt is the PI of. I'm a member of Project 2, which is, of course, the mouse doctors in the group. We also have a Project 1, which studies humans, in particular young girls. In addition, I'd like to acknowledge the support and the actually inspiration for this particular talk from the Community Outreach and Translation Corps, headed by Janice Barlow, Kathy Koblik, Karen Pierce, and Kaya Balik. And finally, I'd like to recognize that this is a collaboration of diverse groups at UCSF, at Lawrence Berkeley National Laboratory, at Kaiser Permanente, the Division of Research, and the Zero Breast Cancer Fund, and that the video itself was supported by a generous donation of Michael Hoff Productions. Thanks.